Welcome everybody. Today I will be talking about DNA replication, transcription, and translation and the different processes that happen. Hope you enjoy. Let's start off with replication. In DNA replication, an enzyme called helicase is used. The double helix DNA strand unravels by the enzyme helicase. The helicase starts from one point on the DNA strand and starts breaking the hydrogen bonds between the complementary bond pairs one at a time, which leads to two different strands. Once the two strands are separated, free nucleotides from the surrounding nucleoplasm bond with the unpaired bases on each of the parent strands. DNA polymerase enzymes help catalyze the bonding between the two bases. Now that the strands are separated and nucleotides have been added to the strands, the next step is transcription. During transcription, a section of DNA produces mRNA, which is also known as messenger RNA, the smaller section of RNA that carries information for protein synthesis. A DNA gene can be seen unraveling. Also, RNA polymerase is seen. There are three nucleotides that start bonding from the surrounding nucleoplasm, using one of the two DNA strands as a template. RNA polymerase comes into the scene as it is used to catalyze the bond. RNA nucleotide is made of a phosphate group, a ribosugar, and a nitrogenous base. The four bases of RNA nucleotides are adenine, which bond with uracil, and guanine, that bonds with cytosine. Part of protein synthesis is translation. After the mRNA is finished transcription, it exits the nucleus through the nuclear pores and goes to the ribosome, where it aligns accordingly. Then a tRNA, transfer RNA, with a specific anticodon that corresponds with the mRNA codon joins, bringing down a specific amino acid. Second tRNA, bringing a specific amino acid, ends up beside the first amino acid, and both amino acids join together and make a bond. This bond is called a peptide bond. Since the first tRNA has fulfilled its purpose, it breaks its bonds with its amino acid. The second tRNA moves one spot. This allows a space for the third tRNA to arrive with a new amino acid. This makes the polypeptide chain longer. The process stops when the codon on the tRNA tells it to stop. After the polypeptide chain is set free, a protein is made, and the protein can be used in the body.